Hello, today I'm going to be talking about mastering the toxicological risk assessment of your device with better chemistry, and specifically focusing on minimizing risk with confident extractable assignments. My name is Philip J. Furco, and I'm a team lead senior scientist with Eurofin's EAG Laboratories. So in my current role, I support chemical characterization of medical devices by developing new methods and driving process improvement. That's kind of the main thrust of my work, but I do also touch on other aspects of this. So some of it's development of methods in new directions for medical device testing, not just extractables and leachables, but also a lot of project support, especially for exceptional projects in terms of the setup or aims, and just kind of helping to navigate projects smoothly through scientific challenges. So my focus prior to this work has largely been on high resolution mass spectrometry with liquid chromatography separation. And so if my talk seems heavy on that, it's because I'm probably most familiar with it, although my work does touch on all aspects of chemical characterization. My previous experience includes pharmaceutical deformulations and quality control work. So I have some industry experiences that kind of relate to it or not exactly the same thing, which is can be beneficial for reasons that we'll see later in this talk. Before I get into the details of it, though, I do want to say that this talk really reflects the current standard in interpretation, and both of those are evolving. So in 2020, the ISO 10993 Part 18 standard was re-released. In 2022, an amendment was released that had some pretty substantial comments on the calculation of the uncertainty factor. And then CDRH has proposed guidance in 2024 coming out that's guidance on the chemical analysis for biocompatibility assessment for medical devices. So you can see that, in several different ways, the attention being paid to interpretation is changing. So this all should be considered, kind of contemporary to the date at which I'm covering it, because, interpretation is going to change. So overall, what I'm going to talk about are techniques to decrease unidentified and tentative annotations and increase the accuracy of semi-quantitation. And this covers all aspects of the data collection and interpretation process, and we'll kind of follow it through in a chronological order. But the topics that we're going to cover are MSN coverage, APCI to supplement ESI, so these are ionization techniques for LCMS, data deconvolution, data interpretation tools, internal MSN libraries, as well as training. So what we can see here on this slide at the top is sort of a conceptual diagram of the process for generating and interpreting chemical characterization data. So we're not starting at the very beginning, we're kind of starting just upstream of the part that my talk covers, and we're finishing out just a little bit downstream of it. So going from left to right, we have device extraction, extract analysis by analytical chemistry methods, interpretation of analytical chemistry data, and then the generation of the chemical characterization report, and then the last one that we'll kind of consider in this talk, which is the toxicological risk assessment. So what we're really going to focus on are the three in the middle. There's the extract analysis itself, so the instrumental methods, the interpretation of the analytical chemistry data, and that's going to be through a variety of approaches, and we'll talk a bit about those. And then all of that is going to be put into that chemical characterization report. Now, what's important to understand here is that you can generate chemistry data for lots of different reasons, right? And probably the most abstract would be an academic setting where it's for pure science applications, and the deliverables, journal articles, right? But ours, in this instance, very specifically, we're generating data to support a toxicological risk assessment. And the reason that's important is that our audience is a toxicologist. So we need to be sure that we're giving them what they need in order to make an accurate, timely assessment, no more, no less, and we need to be speaking their language. So what does that look like? It's about the analyte information. So the analyte information it includes estimated concentration, identification, and the confidence level of identification. So obviously the identification is some sort of information about what the compound actually is that we're seeing. The estimated concentration is a semi-quantitative approach. So these are different from targeted screens, right? And we'll get into that more on the AET discussion. And then there's the confidence level of that identification, which is sort of a standard and a shorthand way of describing the precision of the data that we're generating. So these confidence levels are important because they affect the toxicological risk assessment, or TRA, through the margin of safety, or MOS. So for a confident annotation, typically an MOS greater than 1 is accepted. But if it's tentative, an MOS greater than 10 is accepted. So this is an order of magnitude shift. And so it's really important that we identify as many things confidently. We identify many things as confident identifications as possible to improve the precision of that TRA. So I'm not going to be able to go into it in detail in this talk, but the confidence levels and their determination are based largely on USP 1663 as of the time of this talk. So next we'll go a little bit into the identification of analytes. So here I have two schematic mass spectra. The one on the left is an MS1, the one on the right is an MS2. So in an LCMS experiment, typically what happens is you have the MS1 on the left, that vertical bar represents some peak that's eluting from the instrument, and then this peak can be isolated and fragmented to give the data that you see on the right. So there's more fragments that can be used for library matches and spectral interpretation. So typically, if you only have data on the left, basically you have some peak coming out and a single ion associated with it, you can relate that peak area to a concentration, so that's our semi-quant, and then you have an exact mass, which can give you a predictive formula. And that level of detail really is probably almost always going to be a tentative level assignment at that point. And so as we know, that's not going to be very helpful. We want to bump that up, right? We want to get to a confident level so that our MOS evaluation is a little less stringent. It's going to be 1 instead of 10, or better than 1. So the MS2 helps with this because this fragmentation process gives us stuff that we can look at and we can compare it to library matches, and that can give us increased identification, or we can do spectral interpretation again for identification. That increases the confidence level. So the big takeaway from this slide is that if you only have MS1 data, you're probably going to be stuck with a tentative assignment. If you have MS2 data plus the MS1 data, you're much more likely to get that confident assignment. So the question is, how do we increase the coverage? We get more of that MS2 data. So one of the ways we can do that is through dynamic exclusion lists. So the image we have here on the right is an instant in time from a chromatographic run. So we've got, it's basically a single mass spectrum, and each one of these vertical peaks is a discrete ion that's reaching the detector at that point in time. So as you can see, there could be hundreds of peaks coming off the instrument pretty much at the same time, and the instrument has limited capacity to collect MS2 data for all of these. So what can be done is programs can be set up to generate dynamic exclusion lists where the time and the individual ions at that given time, once they're analyzed once, they're added to this list and they'll be skipped over the next time those ions are observed. So we can see that on the first injection, we would only capture the things circled with dark blue rings, and these would be maybe 5, 6 different ions that we would observe. If we re-injected this without a dynamic exclusion list, we would see exactly the same thing, same top 5 or 6. However, with that list that's going to exclude those from analysis, we then see the ones that are circled in orange in that second injection. So we're going down in intensity from the first round to the second round. And if we do another injection, we can even get down into the third round. Now, we don't have to do this all the way until there are no more peaks to be observed, because really all we have to do is make sure that we get full coverage of MS2 or better down to the AET. So again, this is a great way that we can shift tentative identifications to confident by trading off this redundancy that we would have if we just collected the first injection information over and over, and increase our coverage by going lower. So it's using the same amount of instrument time, but it's doing it in a more targeted fashion. It can also be used in conjunction with MS3, which is the next thing I'm going to talk about. So the two images on the left and center are the ones that I've already showed in discussing MS1 and MS2. And so those don't really need any more explanation, but then there's this MS3. So the images here, though, for MS1 and MS2 are a little bit different than the ones I showed previously. And it's the scenario where I have something that ionizes really only to form one peak. So if you look going from the MS1 to the MS2, there's a change in the M over Z values. It's a different ion, but we only really see one. And that's not enough to make a confident assignment. It's not a lot of structural information. So what we can do is we can isolate that single ion and then fragment further in MS3. So it gives us additional structural information. We can then take it through the same process of library matching or spectral interpretation, and that can help us to get that better identification. So at this point, we've kind of discussed just method alterations within the ESI method for improving our rate of confident annotations. So now I'm going to move on to some other things that we can do with this to improve it as well. So the next one I want to talk about is APCI to supplement ESI. So ESI is electrospray ionization, and it's an ionization method for LCMS. APCI is atmospheric pressure chemical ionization, which is also a ionization method for LCMS. So down on the bottom right, you can see two images. These are just the source housings for the instrument. And basically, to switch between the two modes, all you do is you simply swap out parts of the housing. So you can use the same instruments, you can use the same chromatographic techniques with some exceptions, and so you can collect this additional data. So the question is, why would you do this? And the short answer is, APCI can show us things that ESI does not necessarily. So when ESI was originally developed, it was developed for mass spectrometry of intact proteins. So it's a very soft, gentle process that can, as the discoverer of the technique described it, you're giving electrospray wings to molecular elephants. So it's a very gentle technique to take these intact proteins into the gas phase. And that's certainly a great accomplishment, definitely worth a Nobel Prize, but there is a downside to this, and that is if something is not easily ionized, it's probably not going to show up very well. So we can end up with is a very weak signal and no MS2 data. And that's where APCI can help. It's a stronger ionization method, and it can ionize less polar materials. And so again, you can get greater coverage on MS2 or MS3 data. Again, with that, you can then leverage that to move from tentative identifications to confident. The other benefit is that with some forethought and planning, you can use the same chromatographic method so that peaks elute at exactly the same time with ESI and APCI, which further supports the quick interpretation of the data. So moving on more to discuss data interpretation, now that we've discussed the instrumental portion, I want to talk a little bit about the type and the amount of data that's being generated by these chromatographic methods in these studies and approaches to interpreting it. So a lot of ENL work currently is moving towards using high resolution instrumentation for all data and all data collection. And the reason this is useful and important is that it increases the specificity and can provide more structural information. But with that does come a cost, and that is that we're generating gigabytes of data per measurement. So what I'm showing on the right here are a total ion chromatogram on the top image, and then on the bottom I'm showing a mass spectrum. So the total ion chromatogram is the overall chromatogram for the run, which is roughly 30 minutes in this case. So within this run, we're collecting MS spectra, several every second. And so what we end up with is a mass spectrum, every few tenths of a second for 30 minutes. And so we end up with roughly 5,000 of these MS spectra that are shown below. So if you think about an individual mass spectrum as a page on a book, which it certainly could be because we could have hundreds of peaks to observe in each mass spectrum, we have a book that's about 5,000 pages long, and that total ion chromatogram is the spine of that book. Now, knowing that, you should probably be aware that the measurements conducted for this can be hundreds of injections or measurements per project. So, we can be talking in excess of 500,000 pages in hundreds of books that have to be gone through and that data interpreted. So I think it's pretty clear that doing a fully manual interpretation of this data is just not going to be able to thoroughly and accurately assess the data in a timely manner. And so what we need to do is we really need to be using data interpretation tools. And the purpose of these is to automate the more repetitive, tedious, and straightforward tasks in interpreting the data. So that's things like alignment, deconvolution, and data interpretation. And we can do that to the limits possible. One of the major benefits in doing this is that we can do automated searches of MSN libraries. So those kind of fall into two categories that I'll talk a bit more about later. There's the NIST or other commercially available libraries, and then there are also internal libraries that a lab may generate. The last comment I have on all of this, though, is that it's still essential to have subject matter experts be reviewing this data to make sure that it is in fact reliable. The automated routines just aren't there to do it in an accurate manner without any external input. Next, I'd like to talk a little bit about data deconvolution, which is one data interpretation technique that I alluded to in the previous slides. So chromatic graphic data can be deconvolved, and it's applicable to GC and LC. And all data deconvolution means is taking advantage of the specificity or selectivity of a mass spectral detector to separate out individual signals and identify multiple components that may be eluting at the same time in a run. This is important because it enables more thorough analysis of the data set. It reveals relationships between analytes, and it can also detect peaks that are hidden in total ion chromatograms or base peak plots. So I have two examples here. I have one on the left and I have one on the right. The one on the left is an LCMS total ion chromatogram, and the large peaks that we see across that form this pretty Gaussian distribution with nice, relatively even time spacing are a large polymer pattern. It's actually a pegylated, a group of related pegylated species. And they all elute in this pretty typical pattern that jumps out quickly to people who are familiar with the technique. But because of data deconvolution, we're also able to fish out these lower peaks that are plotted in red. And we can see in some instances that these may form shoulders on larger peaks, which are difficult to integrate and get accurate areas, which impacts the accuracy of the semi-quantitation. But even worse, some of them are completely hidden underneath the other peaks. It wouldn't be observable at all except for this data deconvolution. So it goes to show that if you're analyzing your data in tick mode or base peak mode, there is the potential to completely miss analytes that may not be the most intense signal coming out at that given time. And signal strength is related to concentration, not necessarily the more important things. What do I mean by this? I mean that something that is a compound of concern from a toxicological perspective could be at a concentration that is relevant, but it would be completely missed in an assessment using a tick mode and would only be caught using deconvolution. The GCMS tick on the right illustrates a different scenario where we do see several things coming out, but because of the relationship between those ion masses and the retention times, we can be certain, or at least fairly confident, that these things are actually all associated with the same feature. And this is important because if we don't process this data and look through and make sure that we assign all sorts of things, these multiple signals into the one compound coming out, we may have false positives. So we could be searching for some sort of structural assignment to something that's actually in-source fragmentation. And that would be not good, because then we could have a false positive where we may be assessing the safety of a device on a compound that's not even there. So one thing to know about this is that there's no free lunch. This more thorough interrogation of the data does take more time. The number of compounds can drastically increase, but it does help with assignments because you can put the correct fragments with the correct analytes. But again, this is all justifiable because without data deconvolution, compounds could be missed, and they can definitely be problem compounds from a toxicology standpoint. Next, I want to talk a bit about internal libraries. I've talked about libraries in general through this talk, and it's been about the benefits of being able to do automated searches against these, followed up by SME review. So commercial libraries do have some limitations. They're not often focused on extractable and leachable work. They're also prepared using different chromatographic or fragmentation conditions, which can change how exactly the spectral look and how well they match with what you may be generating on your particular data set. They also do not include retention time information, except maybe a retention index, which isn't really the same as retention time in terms of confirming identification. or it has a retention time that's for a different chromatographic technique. So retention time will change based on the technique. Fragmentation will to a lesser extent. So really the best way to get good, strong automated matching is to build an internal library. And so with these, you get those confirmed identifications because you're comparing against a reference material. You've got identical chromatography, which gives you the same retention time within a window of experimental uncertainty. And you also get the fragmentation. But the benefit really comes if you consider this in a broader program for collecting and interpreting this data. So on the left, we have kind of a schematic of the identification of reference materials and their analysis or data collection for a variety of activities. So on the reference material identification, you really want to consider a bunch of different things. You want to be looking at like what's in the literature about compounds of interest around the materials that you're analyzing, the chemist input, common identifications, and materials of construction. Use all of these to select your reference materials and then collect data on those under the same chromatographic conditions as you're using in your chromatographic methods. So what can you do with this once that's done? Well, you can determine relative response factors, which can help for selecting surrogate standards. It also helps to demonstrate the general suitability of the method. Relative response factors can also be used to calculate uncertainty factors, which can help lower the UF, which then increases your AET, which is always a good thing. The third thing is that you can build mass spectral libraries, and these will give you quick confirmed identifications by matching retention time as well as MS2 or MS3 data. The last thing I'd like to talk about is the training. So up until this point, I've been talking about sensitive instruments and powerful data processing, and those things are really only half of the equation in this work. A strong cadre of subject matter experts is really needed to review and interpret the data. And what that looks like in practice kind of covers a spectrum. So people come into this field from a variety of different experiences. I've listed some in this image on the right. So mass spectrometry for E&L can pull in people who are trained in analytical chemistry, in organic chemistry, organic, physical, or biochemistry. And each one of these fields has something of benefit to give to the interpretation of mass spectrometry. No one really has a monopoly on the knowledge all around this. And something that, at least in my experience, have benefited from is being on teams with people from diverse backgrounds in education, where they could share their knowledge with each other and get projects done that with a group of just one type of chemist would struggle. So in terms of the training, once you're on the job, since it doesn't overlap completely, it's important to try and continue that and share with each other. So the spectrum is, though, that on the low end, it's purely on-the-job training. It might be the things that you pick up in your individual projects. But I would suggest that there is a higher level of doing this, and it's definitely worth pursuing because of the efficiency gains. And that is to have regular training. So what this can look like is weekly meetings to discuss, contemporary problems, literature, or present case studies. And this is really important for a variety of reasons, right? First, it's the continuous improvement of those SMEs to become better and better at doing this job and share their information. It's also important for generating a more consistent product, and this can be very useful, especially if you're working on a series of related medical devices. It's also a great place to share FDA feedback. So if a lab is getting information on one project about the FDA expectations or another regulatory agency, it's much better to share that than to submit every single project and hear the same feedback. So that's another benefit of this, and it can help address that point I raised earlier about the changing interpretation of the standard. I think the big takeaway here is that the training's important. It should be scheduled and planned, and that should be going on so that people can share their expertise with each other, because there are no graduate programs specifically for E&L work. Another way to look at this training is from the overlap of experiences or on-the-job training with medical devices. So there's the old adage that experience is bought dearly but can be shared easily, and that's definitely the case with this. So if people have previous industry experience in manufacturing, engineering, metallurgy, medicinal chemistry, polymer chemistry, these sorts of things, this information can be readily shared. And so any person approaching medical devices from one of these angles, they may be a strong expert in that, but if they know at least a little bit about the others, they're in a much better position, because then they can identify what type of problem it is. And through the regular training, people begin to know who the points of contact are for those fields of expertise. And so they can take a very difficult problem and go and talk to someone who's an expert in that and get a quick answer. So I think in the training, to consider it, you've got to think about all the various experiences and educations that lead someone to end up in E&L, and you need to really break down the barriers that might exist in an organization to make sure that people are freely sharing that information with each other, because overall that just leads to a more consistent and higher quality product. So I'd just like to wrap up with some conclusions. I just want to remind you that a common aim of chemical characterization is to provide data supporting a toxicological risk assessment. And so the design of the work and the design of the chemical characterization report is really to do that, to support a timely, accurate toxicological risk assessment. I think I've done a good job of showing that all aspects of data collection and interpretation impact the confidence levels for identification. What did that look like? It looked like discussing the initial methods for the data collection, the choice of methods, right? So we might do ESI a certain way, but then we also add APCI in the case of LCMS. Interpretation, we talked about software. We talked about building tools outside of software like databases. And then we talked about the training of interpretation. All of these things impact the confidence levels for identification. And an effective program is really going to push for improvement on all of those areas. And the last one, I would just like to conclude that a multidisciplinary team using purpose-built tools and methods will generate reports that minimize tentative and unidentified compounds. They're going to minimize turnaround times, and they're going to support accurate, timely TRAs. So going way back towards the beginning to that slide I showed, where it was the conceptual workflow, we ended on the toxicological risk assessment. And for the purpose of this discussion, that was a good stopping point. But the important thing to recall in all this work is that the true end result of a quality chemical characterization supporting a TRA is that a medical device that should be on the market is cleared to be safe from a chemical perspective and can make it on the market to improve or save lives. So this concludes my talk, and thank you for your time.